All right. Well, thank you everybody for joining us for today's session. Today we're really, it's our pleasure to have Sydney from the Baker Lab coming to discuss uh, important work that they've been working on with respect to ProteinGenerator, which is a diffusion model um, over sequence space, which is a little different from what we've talked about previously. But anyway, we're very happy to have him here and take it away, Sydney.
Okay, sweet. Yeah. So thank you guys for having me. Again, my name is Sidney Lisanza. Yeah, and today I'll be talking to you guys about sequence-guided multi-state and functional protein design using a RoseTTAFold sequence-based diffusion. Now, one thing you'll note is that the title is different from what was in the preprint. I'm actually not going to talk about the preprint version. I'm going to talk about the submitted version, so you guys get a cool preview. And I think the story has changed a bit because we've had time to kind of sit down and think about the real benefits that being able to do diffusion and sequence space really provides to you. Yeah, and hopefully you guys like the new story. Okay, so what is sequence-based diffusion? So you guys are so as Nick alluded to earlier, most people tend to be familiar with structure-space diffusion, where you take atom coordinates, you noise them, use a denoising network, the most well-known case being RoseTTAFold, and you use that to reconstruct the structure. To which point this gives you some really nice benefits. The main one is being able to guide for specific structural attributes. So for instance, you're trying to make like a symmetric, like C3 or dihedral symmetric type structure, it gives you that intuitive framework. But you can do the very same thing within sequence space, where you just need to formulate a way to represent sequence such that you can corrupt with Gaussian noise. And so we did that, and I'll go into more detail about that later. And then we use RoseTTAFold as a denoiser. And the way we did this is it allows us to get both sequence and structure jointly concurrently. To which point, not only does that allow us to guide for sequence-specific attributes, which is particularly promising, especially considering the sequence activity data sets out there are very much larger than, say, structure to activity data set. And so you get to be able to guide with sequence-specific attributes, but on top of that, the way we formulated, you can also guide with structural attributes. So really opening up the protein design space, which people have not been able to previously do with deep learning methods, but classical methods, of course, that has had time to mature in that domain. So now I'm going to go into a bit more detail about how we're actually doing this. So as I mentioned earlier, we need a way to represent sequences such that we can corrupt intuitively. And so how we chose to do that is very simple. We represent sequences as one-hot tensors. And so essentially, for a given position, there will be a one corresponding to the specific amino acid, which is at that position, but zeros everywhere else. And so within such this framework, we can just go ahead and add on Gaussian noise to that. And so yeah, and so to the left here, we're showing a sequence corrupted with Gaussian noise, with the one hot tensor showing where my mouse is. But we can actually look at what the actual amino acid sequence looks like in a sequence logo. And there you can see like it's unclear what amino acid it is. So there we're seeing complete corruption. And as I also mentioned, we're using a RoseTTAFold, so we also get the structure out. So likewise, the structure, because it's not a real sequence, the structure is a jumbled mess. And so what we do within T steps is we denoise all the way from XT to X naught, to which point when you get to X naught, now you have a coherent sequence structure pair. So you see that the one hot tensor is literally a one hot tensor. And then if you look at the actual reconstructed sequence from the sequence logo, you see it has very defined sequence amino acids. And then on top of that, you see a coherent structure. Now, again, the main benefit of using this diffusion framework is to be able to guide for specific attributes. And so I'll go into more detail about how we do that. So if we look at the bottom section here, we can guide for, say, hard structural constraints, and these constraints being explicit motifs. Say you want to do active site scaffolding or any type of functional motif scaffolding. But then in addition to that, you can also guide for specific secondary structures. And so you input that to RoseTTAFold in addition to the noise sequence at XT. Now, what the network does is it's going to predict X naught, the X naught both for the sequence and also for the structure as well. To which point, how we actually guide for sequence-specific attributes, more so soft constraints, as we'd like to say, is by taking gradients with respect to the sequence, to which point you can multiply that by a scaling factor and then add it onto the x naught prediction, to which point you noise. And upon noising, you get xt minus 1, and you iteratively do this until the end. You get a sequence slash structure that that agrees to whatever constraints you decided to input earlier. And so here is a GIF of a design trajectory. Let's wait until it starts over. So here at the beginning, you see that the sequence is unknown, but as time goes on, it gets more confidence. And likewise, the structure also gets more confident. Yeah, and so it's pretty cool seeing how this goes. All right, so what are the benefits of sequence-based diffusion? Well, one, explicit guidance over sequence-based attributes. And I'll be going over two specific sub-problems we tackled in the paper for this. One, enrichment for rare amino acids, and then two, the generation of repeat proteins. And then more excitingly is being able to explicitly guide over sequence and structure attributes simultaneously. And so the two projects, the two sub-problems we tackled in the paper, it'd be conditional bioactive peptide scaffolding and multi-state design, where multi-state design, by that I mean you have a sequence that encodes for multiple structural states depending on some conditioning information. Okay, so how exactly did we do amino acid enrichment? So how we did amino acid enrichment is by biasing for certain amino acids. So when you have an input sequence to the model, in this case xt, the model is going to denoise and give you an x naught. And from that x naught, we're going to go ahead and upweight certain amino acids we care about. So in this case, we're upweighting for tryptophan. So what we're going to do is we're going to actually literally add on some weight factors to the tryptophan residues and increase their weight. So to which point, when you get to xt minus 1, you see tryptophan is increased. And so this is a benchmark showing that our amino acid bias potentials actually work. And so we did this for rare amino acids, specifically valine, tryptophan, cysteine, histidine, and methionine. And you can see with the bias, the frequency increases to be approximately 20% for generated designs, whereas without the bias, you see those amino acids are not as frequent. And then to see how these designs compare against native sequences, we have a multi-dimensional scaling plot to the right here, and you see that the AA bias proteins occupy their own unique space relative to what the native proteins would actually, indicating to us we're making novel sequences that nature would not sample on its own. And so one of the amino acids is being enriched for cysteine. Now, cysteine is particularly interesting because it lets us interrogate the model's ability to reason between sequence to structure relationship. So specifically, we're only telling the model, hey, we want a bunch of cysteines here, but we don't tell it that these cysteines need to be paired in order to form disulfide bonds, because if they're not paired, then you're going to get aggregation. So we're seeing, can the model reason about this via the training objective it has. And furthermore, cysteine-rich proteins are highly stable and have certain drug-like capabilities, which make them promising for being protein-based therapeutics. So we went in the wet lab and actually tested these designs. And per size exclusion chromatography, we see that the designs are monodispersed per them eluting in the expected elution volume. And on top of that, we wanted to check the secondary structure content of these designs. So we ran them through circle dichroism, and we see they have the expected spectra you'd see from all helical proteins. And then on top of that, if we increase the temperature at 95 C, we see the CD spectra doesn't change, indicating to us that these proteins are stable up to 95 Celsius. But what we're actually interested in, do the disulfide the model predicts actually form? So to interrogate this, we did mass spec studies with and without reducing agent. So with reducing agent, you'd expect the disulfides not to form. So those cysteines would actually have hydrogens, which wouldn't be present when the disulfides are formed. So that mass difference we can actually use to calculate the number of disulfide formed. And we see for the majority of the designs, all the disulfides are formed, which is really exciting to see telling us that the model is able to reason between sequence structure relationship and is able to design functional proteins readily. And so the next amino acid I'll be talking about is tryptophan. Now, tryptophan is interesting, one, because it has certain spectroscopic properties. So in this case, it absorbs UV light. But then on top of that, it's very bulky and hydrophobic. And so the model needs to reason that, hey, I need to pack these into the core. So again, this allows us to interrogate that sequence to structure relationship. And for application, because tryptophan tends to absorb UV light readily, it'd actually be a promising protein-based sunscreen. So in fact, if you look at the types of proteins that are, say, like in your hair, things that are exposed to sun a lot, you'll see that those proteins tend to be very upregulated in tryptophan. And so again, we tested these in the lab, and we see that the designs elute at the expected volume, indicating that they're monodispersed. But then on top of that, we see that the absorbance of these designs is very high relative to designs without a lot of tryptophan, indicating that, I mean, it's just a byproduct of that many tryptophans. But then again, if we look at CD, we see they have expected all helical characteristic. Okay, so then moving on, I'll talk about sequence repeat protein generation. So actually how we made these sequence repeat proteins is actually very intuitive. We literally take the asymmetric unit after the model predicts X naught, and we copy it over N times, say if you want an N repeat protein. And from which point the model, so then the model just, and then from there we noise and we get xt minus 1. And by doing this iteratively, we get repeat proteins. And so one thing to note, we're only doing this at the sequence level. So the model needs to reason that when you have sequence repeats, that must also correspond to structural repeats. So we don't tell it needs to do that, but the model is able to readily do that on its own. And the application for these repeat proteins is pretty intuitive. It lets you build arbitrary size proteins without needing to without needing to overthink things. So say, for instance, you want like something big, you just copy that N times over until you get the size that you want. So we actually made a variety of proteins with varying secondary structure, and we ran them through the normal pipelines, SEC, CD, and we see that they're monodispersed. They have the expected CD spectrum, the expected CD spectra, and then on top of that, they're stable up to 100C. But what's more exciting, we're able to solve the crystal structure of one of our designs, and we see that there's very strong agreement between the design and the actual crystal structure, indicating that our model is able to design structures with atomic level accuracy. Okay, so what I've told you guys previously has been mostly conditioning on sequence-wide attributes, but now we also have the capability of conditioning on both sequence and structural attributes. So we use this capability to actually scaffold sequence motif for functional proteins. And so how we go about this is we take a functional peptide and we scaffold that into, and we scaffold it such that it's caged. So when it's within the scaffold, it's not active. But we want conditional activation. So in addition to scaffolding that bioactive peptide, we also introduce cleavage, protease cleavable site. And so upon introduction of that protease, this will cleave that bioactive peptide, releasing it to do whatever function that it may want, whether that may be lysis, for instance. And so again, how we do that with the model is we provide hard constraints in the form of explicit sequences for the cleavage site and the bioactive peptide sequence. And we task the model of, hey, give me the rest, scaffold it. But then on top of that, we provide secondary structure conditioning information, which is specifically useful in the case where you're trying to have a protease cleavable site, because you want that site to be accessible to the protease, so you want that to be in a loop as opposed to, say, something ordered. And so providing that information, the model readily gives us proteins with those desired constraints. And so this is actually super exciting for us because it allows us to scaffold intrinsic peptide barcodes. So I'm going to explain exactly what that means, but first I'll introduce you to what people typically do in functional assays with barcodes. So so in this case, these people have a library of antibodies and they want to screen for binding against an antigen, but they want to do that in a high throughput fashion, with strong guarantees that they can get the right functional thing. And so how they do that is they generate a library, and each library has an associated peptide barcode. And so what they need for this is they actually need to generate multiple degenerate barcodes for each specific design, because that barcode oftentimes it can be not well behaved. By not well behaved, then I mean that it causes misfolding of the design or interacts with the antigen in an unintended way. And it just provides a variety of different roadblocks. And so how people overcome these roadblocks with these extrinsic barcodes is by having multiple barcodes per design to, say, account for any confounding variables. And so they introduce these barcodes and they do functional screening per SEC. So how they're able to do this is if the antigen doesn't bind, it's going to be at a lower size. And so it's going to elute earlier. And then if it's just the free antigen, that's going to elute a little bit later than the first fraction. But then if you both have the binding of the antigen by the antibody, then they're going to elute later. Sorry, they're going to elute earlier because they're bigger. To which point you can take each of those fractions coming out of SEC and then you run them through mass spec, to which point you can pinpoint the functional design dependent on the elution fraction. But as I mentioned, you need to have a lot of degenerate extrinsic barcodes in this case to account for any confounding variables for an unwell-behaved barcode. So what would be more enticing if the barcode is actually part of the design. So rather than the design being separate for the barcode is literally intrinsically part of the intrinsically part of the design. To which point you circumvent the need for many degenerate extrinsic barcodes. And so we did that. So what we did is we took a bunch of experimentally verified barcodes. We tasked the model with scaffolding them into the design. And then we ran all those designs in a pooled fashion through SEC, collected individual fraction volumes, ran them through mass spec, and then reconstructed what that SEC trace would have looked via mass spec. But then in parallel to this, we individually expressed all those designs and ran them through SEC. And so we compared the controls being the actual verified SEC traces versus the mass spec reconstructions, and we saw if there was agreement. And so here are example designs in SEC traces. And so the dotted line is the pooled library, and we can see that the barcode and individual SEC traces agree with each other, and there you can actually deconvolve them from the pooled design. But then if we look this at the more global scale, where we look at the peak volume difference between the barcode reconstruction and the individual SEC traces, we see fairly good agreement, with differences between the peak volumes being less than 1 mil for most of designs. So we believe that this is a promising approach for functional assays, for high throughput functional assay screening. Okay. And then, so the next thing we also wanted to see is, can we scaffold bioactive peptides? In this case, the bioactive peptide is melittin. Now, melittin is interesting. It actually comes from bee venom. And what melittin does, it actually lyses cells. And so, for instance, say you want to kill cancer cells. If you could find a way to cage melittin such that it's only released upon the present in say a tumor microenvironment, then that's a viable way to do targeted cancer cell destruction. So how did we do this exactly? So what we did is we took melittin, scaffolded it into a protein via our model, and then we introduced a furin cleavage site. Now, furin is interesting because furin is a protease that is upregulated within cancer cells. And so we put that at the end, connecting the melittin to the scaffold. And so the idea being, we should be able to cleave this and have conditional cell lysis. And so we took this design, we ran it through SEC, and we see there's monodispersed species. So we took that monodispersed species, ran it through SEC, and then from there we see that it's has expectancy characteristic for an all alpha protein. But now we want to see, can it actually be cleaved by furin? So we actually we ran the design, we did the reaction, and did the gel for it. And so the design is here, and you see it's larger than, say, melatonin. But then if we introduce furin, you see now there's the introduction of two species, the uncleaved design and also the cleaved design. But then if you look here, it's a bit hard to see, but there's also a band for melatonin, indicating to us that that yes, this is furin is able to indeed cleave and release melatonin. But we went a bit further too, because we actually wanted to see is can we actually do conditional hemolysis of cells. And so to the left here, we have our positive controls, Triton X100, which lyses cells, melatonin, alone, normal melatonin. And again, we see both those lyse cells. But if we just have buffer, we see no lysis. If we just have furin, again, we don't see any lysis. And with just the design, we see no lysis, indicating to us that our design is in fact caging melatonin. But then if we both introduce furin and the design, we see that there's conditional hemolysis. So that's really promising to see. And this is the first step towards making conditionally active tumor degrading designs. Okay. Okay, so the next thing we wanted to see, so a really interesting problem within protein design, which has been really intractable for the vast majority of the fields, timeline of the field, is being able to design proteins that can account distinct structural states just from one sequence. And the reason you'd want to do this is, again, it gives you a way to do conditional activation. And conditional activation is really promising, say, in the instance where you want the design to be active in some environment, but not active in another environment, like in the melatonin case. And so how we chose to see if the model could address this problem is we decided to model a protein that has a specific structural state within the parent design. In this case, you have an alpha beta protein, but upon cleavage, you get two children designs, and we want those two children designs to be all alpha. And so that was a design objective that we set out to solve. And so how we do this with the model is we run a sequence through the model with three different conditions. The first conditions is in the case prior to cleavage, where you have the full protein, but we task the model that it needs to be alpha beta. And then for the children, we ran that such that the model needs to put it in an all alpha state. And so we ran the same sequence through the model with different conditioning, and the model predicts out X naught. And so we take those X naught predictions, we actually average them and then noise them to get XT minus 1, then iteratively do this for however many steps, to which point we get a sequence that the model believes to adopt distinct states depending on the condition. In this case, the condition being cleavage. So these are example designs where you have the parents having an alpha beta fold shown, and then the children having all alpha shown as well. And so what we did, the first experiment we did is, can CD detect distinct secondary structure content shift in the designs? And in fact, it does, because if you look in the case of the parent, you see the parent has increased beta content upon CD deconvolution compared to the children. But then if you look at the children, you see the children have higher alpha content compared to the parent. But this was promising, but what we really need is actual structural studies so we can actually interrogate what's going on. So we sent our designs over to collaborators at Georgia Tech at the McShann lab and saw if they could go about solving the NMR structure for these designs. And so we were able to solve the NMR structure for the two NMR structures for parent designs. And we see that there's strong agreement between the design shown up top and the NMR ensemble shown at the bottom here. But the problem is, so that's really promising to see. But then in addition, if we look at the actual spectra of the children compared to the parent, we see that the children peaks are are in distinct chemical environments, indicating to us that there might be fold switch occurring. And so we see that both for the child A when you compare it to parent, and also child B when you compare it to parent. And then we see the same thing within this next design, where we see that there, again, atomic level agreement between the parent and the NMR ensemble of that parent. And again, distinct chemical shifts between the child A and a parent, and again, child B with the parent. But we wanted to, it was because it was difficult for us to solve the actual NMR structure for the children, we wanted to see whether we can get any more information from the NMR spectra. And it turns out you can. So it turns out that if you look at all alpha and all beta proteins, they have distinct chemical shift influenced by their secondary structure. So an all alpha, and so in this case, if you look at the plot here, an all alpha protein would be more shifted towards the top right quadrant, and an all alpha protein of reference is shown here in red. And then if you have an all beta protein, you'll be shifted more towards the bottom left. And so in this case, we have an all beta reference shown here. And then if we only pay attention to the family shown in family shown in green here, we see that the parent here is more shifted towards the left, indicating to us that it has more beta content. And then if we look at the children, you can see that they're shifted more towards the right, indicating to us that they have higher helical content. So these results in conjunction tell us that we were able to design multi-state design proteins, to which point the parent has a distinct fold from the children, which is really exciting to see. And it's something we haven't been able to see with current deep learning methods to be able to explicitly design for multi-state proteins. And then another super exciting avenue for a sequence-based diffusion model is to be able to use sequence activity data to guide for higher active designs. And so we did a in silico test of this using the GB1 data set. And what GB1 does is this protein here in gray, and this protein binds onto IgG. And what people have done previously is they've made exhaustive mutations to these 4 sites, and we're able to make sequence to activity data sets, to which point we can easily look up as if we did the actual experiment. And so to test our model's capability to do iterative guidance, we tasked the model with taking GB1 and tasking the model to make mutations to this design, and then taking those designs and then actually looking up the activity, making a sequence activity data set, and then using that sequence activity data set to train a surrogate model to predict activity just from sequence, and then using the surrogate model to guide for generation. So what you want is each successive round of this, you want activity to increase. And so we actually do that, and we see that activity does increase, and our model here is shown in purple. So you see round 1, activity tends to be low, but with each successive round, the mean activity is increasing, which is super promising to see. And especially we're doing way better than the Bayesian optimization baseline shown in gray here. And then one thing to note here is each round has 96 designs. And so in this case, we're in the low end regime, which is really hard to model. And so what we think is happening here is our model has explicit understanding of sequence to structure relationship, which is able to leverage during the design process here to give you more active variants with fewer rounds and fewer examples of designs. So all this work was done with my co-authors here, and they're continuing on the work, and I'll just be highlighting a few of their projects, which they'll be working on to further show stuff. And so the in silico directed evolution results I was showing you would actually be super cool if we can actually apply it to actual enzymes. And that's exactly what Jacob Gershon and Lucas Arnoldt are currently doing. So be on the lookout for their work whenever it comes out. And then Sam Tips here, what he's doing is he's taking the model's ability to be able to scaffold arbitrary sequence motif, whether they may be structured or unstructured, to do T-cell epitope scaffolding, which can be applied for vaccine design. So be on the lookout there for hopefully more vaccines. And then Jeremiah Sims, he's taking this ability to be able to do intrinsic barcodes to do mass spec-based functional assay screening, which will drastically increase the throughput that people are able to test designs and hopefully increasing the capabilities of problems you're actually able to tackle with protein design. Yeah, and so the work I've been presenting here is from the submitted version, but currently we have the preprint out, so go ahead and check that out. And on top of that, we also released our code both on GitHub and also this Hugging Face here. So please go ahead and check those out when you have time. And then, so this was a very big team effort. So big thank you for everyone who worked on ProteinGenerator, specifically Jacob Gershon, Sam Tips, Jeremiah Sims, Lucas Arnoldt, Sam Hendel, Una Yase, and then our collaborators over at the McShan Lab. Because honestly, being able to try and solve structures with such dynamic proteins as we've designed here, that is a very tall order. So thank you for them for putting in the work for that. And so specifically Marianne Sema and Claire Tharp. And then also special thanks to my girlfriend for reminding me I have to give this talk. And then also everyone within the IPD administration, David Baker, Linda Stewart, Lucy Goldschmidt, and Candice Van Warmer. And so are there any questions?
I have a clarifying question just to start, if that's okay.
Yeah, go ahead.
When the model makes a prediction, is it predicting the structure from the predicted sequence, or is it predicting everything all at once?
It's predicting everything at once. Yeah. So yeah, I think it's a common misconception that I've had with people before this, but sorry, I have a lot of slides. Okay, there we go. Yes. So essentially what the model is doing, it has more information when it's giving the sequence because it has the XT from previously, so it can leverage that information to predict to denoise the sequence. But what it's doing when it's getting the structure, it can only go off of what structural motif that is currently present. Or no information at all. But it's akin to whenever language models do like autoregressive, where they go literally from black hole to a like, I don't know, like a character. It's the same thing here, except that you're doing the structure. Because they're happening simultaneously. And if you look at the and if you look at the like GIF here, you actually see that the structure forms before the sequence is completely coherent. Yeah, and so I would say it's the best way to think about it is doing simultaneously, not that the structure comes after the sequence.
Got it. Thank you so much for clarifying.
Mm-hmm.
Yeah, thank you so much for that wonderful, wonderful discussion. I was curious about, so showing this GIF really piqued my interest about like how quickly the structure itself forms into a kind of coherent structure, very globular, like native protein, like before the sequence really determines itself. Now I was curious, have you like investigated how the sequence prediction part mimics maybe protein MPNN or something like that at this later stage when the structure is already really coherent? Have you done any?
Yeah. So my interpretation is that if you look at the space of sequence space versus the space for structural space, um, I would argue that structure space is smaller than sequence space. And a good example to kind of like put that concretely is if you imagine a single fold can have very many like so many sequences that can go to that specific fold. The mapping is very much many to one. And so I think what's happening here is it's easy for the model to be like, all right, we're in this subspace of structural space, so not much is going to change, so we can stick here. But the problem is that even though you have defined yourself within this space, within structural space, sequence space, the possible things are still very big. And so that's why it's kind of taking some time. And yeah, so the later ends, yeah, you could assume it's essentially fixed backbone design at that point, which would be what MPNN does.
Awesome. I have more questions, but I want to open the floor up to anybody else who might have anything.
I'll have a quick question. I really
Mhm.
enjoyed your talk. When you were talking about the peptide, active peptide and cleavage site design, the melatonin system,
Mhm.
can you talk a little bit of the size of the actual backbone that you designed? So not the actual part of the peptide and the protease recognition site, but everything else. Is
Oh,
this
the
important
size?
for the outcome?
Yeah. Sorry. I don't remember the exact size, but I would say it's around 100 residues for the scaffolding.
And can you make it like 10 amino acids or like massive?
Oh, yeah, that's a good question. I think that is something you'd have to interrogate experimentally, how small you can make it. But I mean, just looking at this picture, if you look at let's see. Okay, so you can probably scaffold this with a 3 helical bundle. To which point, I think you need at least a 3 helical bundle if the thing is looking like if the peptide is taking on this alpha helical thing. Because I guess what I'm imagining here is you just really need 1, 2, 3 to kind of provide support to this. But mind you, that this buttressing helix in the back is probably providing a further stabilization. So even though you may be able to do 3 helical bundles, it might very well not be it may not very well be very well behaved. And so maybe it's something you can interrogate a bit more. Yeah. So I guess that's my take. I don't know if I answered your question though.
Oh yeah, no, that's totally fine. Thank you.
No problem. All right, Nick, what are your questions?
Yeah, so one kind of building off of this one about the scaffolding, the bioactive peptides. Um, I was curious. So when you kind of like showed out the formulation for the design, how you kind of went from the sequence with the DSSP kind of guidance for it.
Mhm.
Um, I think it showed, like in your slide, it showed that it was mostly scaffolding with helices. I don't know, have you tried anything other besides helices? I know helices are relatively simpler to design, but yeah, like
Yeah.
structural
Yeah.
conditioning.
So okay, so to be perfectly frank, this isn't super accurate because we didn't specify what secondary structure the peptide sequence should be. And so the model just reasoned that something helical would be good.
I see.
But I will say that like what it chooses is dependent on the actual peptide sequence. Because I guess a good example is if we look at the barcodes. So here it shows for whatever reason that it's best to scaffold this within a strand, rather than a helix. And here it decided to do like a kinked helix here.
Right.
So what the model chooses is dependent on what the actual sequence of the peptide is. And maybe it's reasoning, I don't know, maybe it's doing it smartly, like, oh, the amino acids here, they are more likely to fold to adopt like a helical or beta sheet fold. And so maybe if you do force it, the model can think of some buttressing amino acids to be able to make it conform to whatever thing that you want. But it's something that we haven't interrogated, but could be worthwhile given if like a designer has whatever objectives that they care about.
Cool. Yeah, that's really cool to see and hear that like you weren't actually specifying secondary structure for the peptide and the rest of the scaffold as well. That was really cool. Um,
Mhm.
so I have a more like technical question based on like the diffusion framework that you used for the sequence itself. Um,
Yeah.
so I'm familiar with like discrete diffusion as in like D3PM. I don't know if you've read that. But yeah, so
Yeah,
that
yeah, that's what the transition matrices, yeah.
right. Yeah, that frames it in a very different way rather than like this kind of continuous Gaussian diffusion kind of framework. So I was
Mhm.
curious why you ended up settling on this framework. Have you tried the other ones?
Yeah,
And
so
yeah,
the main thing is you got to think about guidance. How are you doing guidance? Here it's very intuitive. You literally add the gradients and you get yeah, and you're able to go towards whatever direction you want based on the gradients. And if yeah, and then you're talking about the Hugenbond paper. I don't know how to pronounce his name.
Yeah, so there's that one I think that introduced like the binary diffusion, but then there's an Austin paper that was released from the Google research team.
Yeah,
That
yeah. So there they didn't provide an intuitive way to do classifier guidance. And classifier guidance is super important to us, specifically when you have, say, like a surrogate classifier that goes from sequence to activity. So we need that capability. And so in their framework, it's perfectly fine if you have a region where classifier free guidance works. But in the case with proteins, we don't have enough data set for that. And so we really need to be able to guide explicit classifiers for the given functional proteins that we care about. But I'm sure like some math kids out there are going to figure out how to do it properly with guidance in such a way. But when we made the method, that framework wasn't amendable for what we cared about at the time. But I'm sure in the future it'll come out.
cool. Yeah, I think that totally makes sense. The classifier guidance is super important and really enabled a lot of the applications that you showed. Yeah.
Mhm.
Any other questions from anybody else?
I have a technical question on this slide as well.
Yeah.
Just curious, is there a reason why it predicts X naught as opposed to predicting the noise component and calculating X naught like some of the image generating diffusion models do?
Yeah, so I think what it comes down to is what works empirically. And what we found is predicting X naught works more empirically than compared to predicting XT minus one, for instance. And also, just one thing to note is that if you're predicting X naught, the losses that you're able to use are a bit more intuitive, for instance, right? Because in this case, we're using CCE rather than an L2 loss. And we wouldn't be able to use CCE if, say, we're predicting the noise, just because the distribution that we're comparing against wouldn't be like easy to implement per se. And then especially what we found within working within categorical space, CCE is really just an excellent loss that seems to be able to capture a lot of the learning better than, say, using L2 and other things.
Got it. Thank you.
Hello. Thank you for the great talk. Lots of exciting applications. Can you hear me?
Yes, now I can.
Okay. I am working also on a discrete diffusion model, so I wanted to ask if you saw a relationship between the KL loss and the time step that was assigned to like a certain example during training.
Can you ask that question again? Did I see a relationship between the KL loss and what again?
The time step that you assigned to like that protein that example during training.
Oh, are you asking the KL loss explodes for certain time steps as opposed to others?
Yes, if there's like a linear or like what kind of relationship if you know.
Yeah, so actually it's been a while since I've looked at that benchmark. There was some weird stuff going on. And I think what happened in our case is that the loss was weighted low enough to the point where when weird stuff does happen, it didn't influence training too much. But yeah, no. I definitely remember weird stuff happening. I'm sorry, I just don't explicitly remember. What are you seeing in your case though?
Because I was expecting there to be like as the if like for lower time steps assigned to that example, I would expect a lower KL divergence. I don't know if that's quite accurate, but it's not like a linear relationship.
Yeah, okay, so you're
There's
seeing the
some
loss is
correlation.
higher at lower time steps?
Sometimes I
Yeah.
think it is lower. It's like lower at high time steps and lower at um low time steps, but in the middle is like I think it's like kind of parabolic.
Yeah. Yeah. Sorry, I wish I remembered. But yeah, no. Yeah, it's weird. Honestly, there might be just some bug, so you could like, I don't know, just try and figure out if something's like that. But yeah, no, I remember KL being weird. Sorry, I can't provide any more info.
Yeah, thank you.
Well, I
I
have
have one.
another question. Oh, sorry, you go ahead.
No, go ahead. Sorry.
Oh, well, my question was kind of based on something I read in the preprint in the methods section.
Mm.
You mentioned that you during the training, um, you weren't always tasking the model with the diffusion task itself. You
Yeah,
also had a couple other training objectives,
yeah.
including like the traditional given a sequence, what is the structure, and vice versa, the inverse folding problem as well.
Mm.
I was curious how you trained on those. Like, for example, um, when you're like giving your inputs to the model itself, say we're doing the protein folding problem. So
Mm.
we have the sequence itself. What are you, for example, giving for the time step? Like, are you saying that we're at a low time step? We've already decided on the sequence. What is the structure? I'm very curious to see what you did with the time step embedding for these alternative tasks.
Yes. So say we're doing structure prediction, then the time step will be 0 because the sequence is fully defined. And then say we're doing inverse folding, then honestly, the time step should vary. But I think we did something in the outputted one where it wasn't, sorry, the actual release code when we didn't let the time step vary. So I think in those cases, it's always it has to be, yeah, it has to be XT. Yeah, it's always big T. But to be honest, what we're currently doing and what we should have done back then is that T should be varied. That T should have been sampled, to which point you can do fixed backbone like sequence design, but with being able to guide with like whatever gradients that you want. And then let's see, do we have anything, another task I'm forgetting? Or I think those are the three main tasks. Just
Yeah, I think those were it. And then there were like extra conditioning information based on.
yeah. So it's interesting because yes, because we did all of that, theoretically the model can do inverse folding, structure prediction, and generation. Obviously, it doesn't do all three of those well. But yeah, but it's interesting how general these methods can be.
Do you think this model would be able to do a task like designing a globular protein with a large hydrophobic moment where it has to be biased towards hydrophilic amino acids in one region, towards hydrophobic ones in another region, but the regions are separated not in sequence space, but only in structure space?
Mm, that'll be interesting. And I'm guessing you don't mean fixed backbone design.
Okay.
Wait, sorry,
Thanks.
sorry. I'm asking, do you think in your case, would this be for fixed backbone design where the structural is held fixed, or are you imagining that you're also generating the structure as you go?
I was imagining from scratch. I was just curious if that would be possible. I think if in the case of fixed backbone design, yeah, probably. But I'm curious if it could be done without the fixed backbone using a model
Yeah.
like this. Do you think there'd be a way to do that?
Yeah, let me think. The only way I can think about it is if you held some coordinates fixed. So say for instance you do like the motif doesn't even have to be real, but you can put motifs on the N and C terminus. And then go ahead and bias those motifs to be hydrophilic or hydrophobic on both ends, and then let the model reason about that. I don't know how well that would work, but that would be like, I guess my first step towards that.
Okay, thank you. And really nice talk. This is so cool.
Thank you.
Do we have any other questions?
I can probably think of one more if that's okay.
Yeah, go for it.
So, do you think, considering that this model has control in sequence space, do you think there'd be a way to make it generate a protein that has like a disordered region in a specified region of the sequence, maybe by like controlling the model to maintain low structure confidence in that region or something?
Yeah. Okay. So disordered proteins. Okay. I don't entirely understand the biophysics. So I mean, you could specify that that region be a loop, via DSSP conditioning. But the problem with that is oftentimes loops are actually super structured. They
Yeah.
oftentimes have hydrogen bonds connecting them.
Right.
Um, so as is, I think the closest thing you can do is having a loop there. But as you said, you could also have a classifier. Oh, no, yeah. Okay, so you can classify loops, or if you have some sort of classifier that predicts a region is disordered or not, you can use that classifier to explicit like certain regions to be disordered. So you could do that. So those are kind of like your two avenues. But the first one you can easily do, but the problem is that loop might actually be structured. And then the second one is it's entirely dependent how good that classifier is. So for the studies we've done, it's the model is able to lower the classifier, any arbitrary classifier score. But if that classifier isn't very good, then even though the classifier score is going to like improve for whatever thing you want, it might not actually viably look like anything useful just because it's like the model has figured out some space which the classifier is unclear about. Like a good example of that is in the preprint version, we actually took it out of the submission version, is we used a Go classifier to guide towards specific function, but the Go classifier is actually really bad. And so even though the classifier scores increase for the specific thing that we care about, if you actually look at the structures coming out, they don't look like that given thing that you care about. So those would be the two pitfalls.
I see. Thanks so much.
Mm-hmm.
All right. Well, if there are no final questions, then please everybody give Sydney a wonderful round of applause for the very, very great presentation that we had today.
Yeah. Thank you
And
for
I look
having me.
forward to seeing you all in 2 weeks where we have our next presentation. Thank you all for coming. And thank you, Sydney.
Okay. Bye.